A warm good morning to one and all. I am Ashwin Nath of 63rd batch. Today we are going to discuss the topic copper metabolism and its related disorders. Now we will be dealing this topic under the following headings: Introduction, the food sources and the daily requirement of copper, distribution of copper in the body, metabolism of copper, the biochemical functions of copper, and the disorders of copper metabolism. In humans, even though copper is present only in milligrams, it is essential to the proper functioning of organs and metabolic processes. Although Hippocrates is said to have prescribed copper compounds to treat diseases as early as 400 BC, still now scientists are uncovering new information regarding the functions of copper in our body. The free copper present in our body is only 4 to 5 percentage, and 95 to 96 percentage of it is bound to the ceruloplasmin present in the various tissues of our body. Now the total body copper content is 100 milligram. The daily dietary requirement is 1.5 to 3 milligram per day. Now the copper occurs majorly in muscles, bones, brain, heart, hair, kidney and liver. The dietary sources of copper are cereals, meat, liver, nuts and green vegetables. Now various enzymes playing important role in the various metabolisms in our body include ceruloplasmin, tyrosinase, cytochrome oxidase, ALA synthase, superoxide dismutase and phenol oxidase. Now copper is also present as part of proteins in various organs of our body. Like in brain it is present as cerebrocuprine, in liver as hepatocuprine, in RBC as hemocuprine, in bone marrow as erythrocuprine and as the oxygen carrying blue pigment in hemocyanin. Now the names of these proteins need not be by hearted. We have to just remember these names by remembering the tissue or the organ in which they are present. Now let us see the metabolism of copper that we take in through our food. Now the daily requirement is 1.5 to 3 milligram. Now taking its average to be a 2 milligram of copper is taken by us through the food. Let us see what's happened to it. Now the 25% of this copper that we take in is not at all absorbed and is lost directly into the stools. Out of the leftover 75%, 50% forms a complex with a protein called metallothionein and is also excreted through the stools. Now the 25% of the left copper is transported to the liver and is incorporated into a protein called ceruloplasmin, which is transported through the blood plasma as well as it is excreted through the stools, through the stools reaching by means of bile. Now most of the copper present in the liver is excreted via bile itself. Only less than 5% circulates in the serum and largely getting bound to ceruloplasmin, and a very small fraction occurs as free serum copper. And the excretion of copper through urine is also negligible. Now here are some of the food material that are rich in copper. These include beef liver, dried apricons, mushrooms, oysters, crab, hazelnuts, cashew, dark chocolate and sesame seeds. Now let's see the copper distribution in our body. Now as we know, if the dietary copper is 1.56 milligram per deciliter, most of the dietary copper is excreted and nearly 1.5 milligram per deciliter of the copper is excreted as in the fecal matter. Only 0.06 milligram of the copper is utilized. Out of this, a major fraction of the copper is present in the skeletal muscle, the skeletal bones, then present in the muscle and then in the liver, binding with the ceruloplasmin, and a very small fraction is present in the plasma. This is excreted. A small amount of this is excreted as urine as a urinary fraction, and the left as the sweat. Now the recommended dietary allowance of copper is determined by a variety of indicators like the plasma copper concentration, the serum ceruloplasmin activity, superoxide dismutase activity present in the RBCs, and the platelet copper concentration. Now here is the dietary requirement according to the different age groups of humans, and differentiating with respect to males and females. Now here it is again a flow chart representing the metabolism of the copper. Now the copper is taken up or absorbed from the upper part of the intestine. The intestinal P-type ATPase present in the intestinal mucosal cell helps in the incorporation of the copper into the albumin first, from which it is exchanged with the transcuprin, which is the copper transporting protein. From there it reaches to the liver tissue as well as to most of the tissues present in the body. In the liver, this copper gets binded with the ceruloplasmin, and it also it has a high catalyzing power of various enzymes like the superoxide dismutase, which plays an important role in the free radical scavenging system of our body, cytochrome oxidase, which is the fourth complex present in our electron transport chain, hence a very important role in the energy production of our body. It is incorporated into metallothionein, and it is getting incorporated into ceruloplasmin, which as we all know is an acute phase protein as well as a transport protein which transports amino acids as well as sugars. Now this copper bounded ceruloplasmin is transported by the blood into various parts of our body like the skeleton, bones, including the bone marrow, brain, muscle, and connective tissue as well as other tissues. It is also present in hair, which helps the melanocytes in the melanin secretion. And then it also, the ceruloplasmin, the other name of ceruloplasmin is peroxidase, which is which has a peroxidase activity. That is, it oxidizes ferrous into the ferric form, which is then bound to the transferrin, and which gets transported in the whole blood into the bone marrow cells for the incorporation into the RBC. Hence it also has important role in the RBC production as well. And then finally it is excreted through minute quantities excreted through urine, majority being excreted through the feces, through the bile, from the bile into the feces. Now let's just take a short recap. It is absorbed from the upper small intestine, and only 10% of the dietary copper is absorbed. Absorbed copper is then transported to the liver, bound to albumin, and exported to the peripheral tissues, mainly as bound to ceruloplasmin, and a very lesser extent to albumin. The metallothionein is a transport protein that facilitates copper absorption. Now the compounds like phytate, zinc, and molybdenum decreases the copper uptake. Now the plasma copper level is 70 to 140 microgram per deciliter, and mostly copper is bound to ceruloplasmin, which is 95%, and a small fraction is loosely bound to albumin. Now the plasma ceruloplasmin level is 25 to 50 milligram per deciliter, and the major excretion of copper is through bile into the feces, and the urinary excretion is very small under normal conditions. Now let us look into the biochemical functions of copper. Now the first and foremost function is in the energy production. The cytochrome C oxidase, which we all know is a part of the fourth complex present in the electron transport chain, plays a critical role in the cellular energy production, and this is a copper dependent enzyme. By catalyzing the reduction of molecular oxygen to water, cytochrome C oxidase generates an electrical gradient used by the mitochondria to create the vital energy storing molecule. Hence, copper has a very important role in energy production of our body. Now, the second function is the connective tissue formation. Lysyl oxidase is another cuproenzyme or the copper containing enzyme which is required for the cross linking of collagen and elastin, which are essential for the formation of the strong and flexible connective tissue. As we all know, this collagen and elastin are structural proteins importantly present in our extracellular matrix, which gives its strength. So the action of lysyl oxidase helps maintain the integrity of the connective tissue in the heart and blood vessels and also plays a role in the bond formation. Now its third role is in the iron metabolism. There are four copper containing iron enzymes known as multi-copper oxidases or peroxidases. These enzymes have the capacity to oxidize the ferrous ion into the ferric ion. This form of ion is then loaded onto the protein transferrin for the transport to the site of red blood cell formation, that is to the bone marrow. Now the ion immobilization from the storage site is impaired in copper deficiency, as copper plays an important role in supporting the multi-copper oxidases. Hence, iron toxification is a major symptom or clinical manifestation in the case of copper deficiency. Here, as in this equation, as it is shown, the iron in the Fe2 plus state, that is in the ferrous state, is oxidized by peroxidase into Fe3 plus state, which is then incorporated into the transferrin. Now, this oxidation of ferrous is helped by the self-reduction of Cu2 plus present in the ceruloplasmin into the cuprous ion Cu plus state. Now, its role in central nervous system. A number of reactions essential to the normal functioning of the brain and nervous system are catalyzed by the cuproenzymes. These processes include, first one is the neurotransmitter synthesis and the second one is the formation and maintenance of the myelin. The now neurotransmitter synthesis include the dopamine beta hydroxylase enzyme. This is very important catalyst for the conversion of dopamine to the active neurotransmitter, the norepinephrine. And this enzyme needs activation by the copper for its proper functioning. Now the formation and maintenance of the myelin. Now, as we all know, myelin sheath is made up of phospholipids and plays a very important role in increasing the conduction velocity across the neuron axon fibers. And the synthesis of this myelin is dependent on the cytochrome C oxidase activity, which is again a cuproenzyme or a copper containing enzyme. Now its fifth function is in the melanin formation. Now again another cuproenzyme or the copper containing enzyme, the tyrosinase kinase is required for the formation of the pigment melanin. Melanin is formed in the cells called melanocytes and plays an important role in the pigmentation of the hair, skin and eyes. Now the next function is in the regulation of the gene expression for detoxification of the reactive oxygen species. That is, a number of signal transduction pathways are activated by copper in response to the oxidative stress in the body and can lead to increase in the expression of genes involved in the detoxification of the reactive oxygen species and helps in more or the proper functioning of the free radical scavenging system present in our body. Now the next function is the function of copper as an antioxidant. Now as we all know, superoxide dismutase is an part of the free radical scavenging system or the P450 enzyme complex present in our body. Now this functions as an antioxidant by catalyzing the conversion of superoxide radicals to hydrogen peroxide, which can subsequently be reduced to water by other antioxidant enzymes. Now the two forms of SOD containing copper are the copper or zinc superoxide dismutase, which is found in almost all the cells of the body, including the RBCs. And the second type is the extracellular superoxide dismutase, which is a copper containing enzyme found at high levels in the lungs and considerably low levels in the plasma. So these two enzymes contain copper, and copper is very essential or inevitable for the functioning of this enzyme. Now, binding of the copper to ceruloplasmin is very important because free copper and iron ions are powerful catalysts of the free radical damage. By binding of the copper with the ceruloplasmin as well as the iron with the transferrin prevents the overactivity of free copper ions as well as iron ions from catalyzing the oxidative damage. The peroxidase activity of the ceruloplasmin, that is oxidation of the ferrous ions, facilitates ion loading onto its transport protein transferrin and may prevent free ferrous ions from participating in harmful free radical generating reactions. Hence, its function as an antioxidant in our body is inevitable. Now, let's go on to the disorders of the copper metabolism. The first one is aceruloplasminemia. This is a congenital disorder in which the levels of ceruloplasmin in the blood in our body is very low. Hence, due to improper iron utilization, this leads to iron toxication in our brain, liver, as well as the pancreatic islet cells. It results in manifestation of various neurological symptoms. Now, the second one is the cardiovascular diseases. Now, as we all know, copper is a constituent of the lysyl oxidase enzyme, which helps in the formation of cross links in the elastin molecule by oxidation of 4 lysine together to form a desmosin molecule. In the absence of copper or during the copper deficiency period, the elastin becomes abnormal due to the absence of the cross linkages in the chain, leading to weakening of the walls of the major blood vessels of our body. And this leads to aneurysm or weakening of the vessel wall of the aorta and its fatal rupture. Hence, it can also cause fibrosis of the myocardium, as elastin and collagen are also important structural proteins in the myocardium, leading to even cardiac failure. Now, what happens if copper is taken in excess, or the copper toxicity? The excess copper intake, that is more than 3 mg per deciliter, lead to toxic infestation of copper in our body. Copper can oxidize proteins as well as lipids. As we all know, copper is a powerful activator or catalyst of the free radical generating mechanisms. Hence, it enhances the production of free radicals. The chronic toxicity of copper is manifested as diarrhea, blue-green discoloration of the saliva, and hemolysis, hemoglobinuria, proteinuria, and renal failure. The hemolysis is due to its role in the incorporation of iron into the RBC, then hemoglobinuria, then in proteinuria, as copper binding to ceruloplasmin can only result in a functional ceruloplasmin that transport protein as well as amino acids, and ultimately in the renal failure. Excess intake of copper will induce synthesis of metallothionein found in the liver, kidney and intestine, which binds copper, zinc, cadmium and mercury so as to make them non-toxic. Now let us see what happens, or what happens when copper is deficient in our body. Now this copper deficiency or insufficiency can result from malnutrition, malabsorption, nephrotic syndrome, or excessive zinc intake. As we all know that zinc reduces the intestinal absorption of copper into the blood and can be acquired or inherited. That is, the deficiency of copper can be acquired or can be inherited. Now copper is said to be deficient in our body when the serum plasma level of the copper goes below 0.7 milligram per deciliter. The symptoms include deficiencies in blood cells as it affects the bone marrow, and bone and connective tissue abnormalities as it affects the cross linkages in elastin and collagen, as well as in various neurological symptoms, as copper is very essential in the functioning of the central nervous system. Now the clinical manifestations include neutropenia, that is decreased number of neutrophils, and second one is the microcytic normochromic anemia. Third one is osteoporosis and bone and joint deformities due to impairment in copper dependent cross linking of the bone collagen and connective tissue. Decreased pigmentation of the skin due to depressed copper dependent tyrosine kinase activity, and then neurological abnormalities due to the depressed cytochrome oxidase activity. Now the next important disorder is the Menkes syndrome or the kinky hair disease. This is a rare disorder and is sex-linked recessive disorder. This is caused by the mutation of a gene coding for copper binding P-type ATPase in the intestinal mucosal cell, which helps in the transport of copper from the intestinal mucosal cell into the blood. A defect in the transport of copper into the blood from the intestinal mucosal cell is induced due to the mutation of the P-type ATPase. This leads to decreased intestinal absorption of the copper. Hence, it is also possible that this copper binds to the metallothionein present in the intestinal mucosal cell, causing toxicity over there. Clinical manifestations include decreased copper in the blood and urine, normochromic microcytic anemia, depigmentation of the hair, growth failure, mental retardation, vascular defects as there is lesions in the vessel wall due to collagen and elastin absence. And now these are the manifestation of this Menkes syndrome. Now the next disease is the Wilson's disease. It is a rare genetic disorder resulting in the hepatolenticular degeneration. This is an autosomal recessive disorder. Now the biochemical defect in this disease is that the gene called ATP7B is a gene coding for the copper transporting P-type ATPase predominantly expressed in the liver. This gene gets mutated, leading to defect in the transport of copper as well as in the secretion of the ceruloplasmin from liver. This results in accumulation of copper in liver and in other peripheral tissues of our body. This disease is very fatal unlike others and results in early death of the individual. Now the manifestations of the Wilson's disease are seen in liver, bone, in heme, in central nervous system, in eyes, in heart and in kidneys. In liver, it causes hepatomegaly, jaundice, acute hepatitis, then pulmonary hepatic failure, portal hypertension leading to bleeding varices, and then finally to the liver cirrhosis. In bone, it causes arthritis as well as rickets. Then it leads to hemolysis, and then in the central nervous system, it results in deterioration in the school performance, behavioral changes, in coordination, that is in the handwriting gets deteriorated, resting and intentional tremors, dystonia, dysarthria, excessive salivation, mask-like faces, and then dysphagia. In the eye, it results in case of Fleischer rings, that is, it forms a brown ring around the iris due to the deposition of copper in the corneal edges. And results in cardiac failure as well as it results in the proximal renal tubular dysfunction due to copper intoxication. Now the clinical manifestations are the copper is deposited in abnormal quantities in liver and the lenticular nucleus of the brain basal ganglia. This leads to hepatic cirrhosis as well as brain necrosis. The low levels of plasma copper and ceruloplasmin is also seen. Increased excretion of copper is seen in the urine. Then copper deposition in the kidney causes renal damage or failure. This leads to increased excretion of amino acids, peptides, glucose, as well as hemoglobin in urea. Hence, symptoms like hemoglobinuria, glucoseuria, etc. Intestinal absorption of copper is very high, about 4 to 6 times, which again increases the toxic copper manifestations. Now, the causes of Wilson's disease are failure of the ceruloplasmin synthesis or the impairment in binding of copper to this protein, or it can be both. Copper is is free in the plasma and hence can easily enter various tissues like liver, brain and kidney and bind with the proteins over there, gets deposited over there and hence results in toxicity leading to its accumulation in liver, kidney, brain and eyes. The albumin bound copper level can either be normal or can be increased. It causes various neurological symptoms and liver cirrhosis, renal tubular damage, case of Fleischer rings, which are the brown pigments around the iris due to copper deposition. Now, the clinical signs and symptoms include the patient's presence in front of you with hepatitis, acute hepatitis and liver cirrhosis, severe hepatic failure, hepatic decompensation associated with ascites. The picture of which is shown below. It also results in peripheral edema, hepatic encephalopathy, and neurological presentation include dystonia, incoordination, as well as intentional tremor. There will be psychiatric presentation, loss of emotional control, aggressive and antisocial behavior, as well as depression. Now, the ocular lesions include the formation of the Kayser-Fleischer rings as well as the sunflower cataract. Now, the Kayser-Fleischer ring is due to the copper deposition in the Descemet's membrane of the cornea, and the sunflower cataract is due to the copper deposition in the eye lens. Now, the laboratory diagnosis includes the presence of Kayser-Fleischer ring. Ceruloplasmin level will be less than 20 milligram per deciliter. Urinary copper excretion will be more than 100 milligram per deciliter. Hepatic copper concentration will be can be assessed by taking a liver biopsy with sufficient tissue reveals presence of more than 250 milligram of copper per gram of the dry tissue. Now, the imaging studies include CT as well as MRI of the brain and the abdomen, which can be carried out to confirm the diagnosis. Now, the treatment include D-penicillamine, which was earlier used but not now presently used due to its toxic nature. Its mode of action is as a general chelator. This causes urinary excretion of the copper. The side effects include fever, rash, aplastic anemia, leukopenia, nephrotic syndrome, thrombocytopenia. And now the presently used drug is trientine. This is less toxic and the mode of action is same general chelator and induces urinary excretion. Now, the side effects is very low. It presents only aplastic anemia and gastritis. So, just taking a summary of what we have seen today, we have started with the food sources as well as the daily requirements of copper and the amount of copper present in our body. Then we considered the copper metabolism, how it goes through various parts of our body and gets excreted. And then we moved on to its biochemical functions, which include in energy production, proper functioning of the central nervous system, in iron transportation, regulation of the gene expression, in the case of oxidative stress, then its antioxidant functions. And then we moved on to the clinical disorders related to copper metabolism like aceruloplasminemia, copper deficiency, copper toxicity, Wilson's disease as well as Henke's syndrome. And we also considered the Wilson's disease in a bit detail as it's a very important topic. Now, I hope that the topic that I took today is clear for everyone, and I am finishing my presentation. Thank you.